our first speaker, Hongzhen Luo, to present the Empower project.
Thank you, Masala. I will share my screen. I hope you see my screen now.
Yes.
Okay, super. Thank you. Thank you, Masala. So my name is Hongzhen Luo. Originally come from China, and now I'm doing the postdoc research at the same team as Masala. Today, as Masala already mentioned, we will introduce the best management practices from Empower project and try to get some inspiration from you and from other speakers as well. Yeah, so a brief introduction of Empower. Empower is a EU-funded Horizon Europe project, and we started last year, January, will last for 4 years, so until end of 2028, with 25 partners from 6 European countries. Our main goal is to reduce nitrogen and phosphorus emissions while promoting circular nutrient management across different key sectors. This project was funded at a very common challenge across Europe, that we see nutrients as essential for agriculture and life, but there are excess nutrient application which accumulate in the soil, air, and water and have led to economic damage and also biodiversity losses. So we really need a smarter and sustainable approach to better manage our nutrients while secure our food safety and the environment. So in Empower project, we have tried to combine technologies and best management practices as well as governance measures to support the circular solutions for nutrient management across Europe and across different sectors. What sectors we are talking about? Here we defined 5 key nitrogen phosphorus emitting sectors, including agriculture, other primary sectors such as aquaculture, grassland, forest, and mining sectors, water and waste management sector, energy and transport sector, as well as industrial sectors. So we have 4 regional clusters in the project, namely Ireland, Spain, Finland, and Belgium. Each of us established 5 multi-actor transition groups. So there is one transition multi-actor transition groups in each sector of each regional cluster, meaning so in total we have 20 multi-actor transition groups. And the activities of the project are divided in 2 parts. It's aiming for 2 approach of outcomes. So the first part is the research part. Firstly, we focus on the nitrogen and phosphorus flow analysis, and then try to build a nitrogen and phosphorus budget to check the current and improved flows of nutrients within the system. So for this research, UGent is leading this task. Meanwhile, we have identified 6 innovative technologies to recover and reuse nitrogen and phosphorus from different sectors and convert them into 8 recovered fertilizers. Demonstrations are now ongoing in the regional cluster of Spain. This will lead to a specific practice innovation that can guide stakeholders to use the circular nutrients pathways. In the meantime, we also try to identify best management practices across all the Europe, particularly focus on our 4 regional clusters to guide our stakeholders how to use the advanced knowledge to manage the nutrients. Based on this, and plus there are also governance measures to guide people how to use the practices and at a higher level of managing the whole sector, the across sector management. So these are the main outcomes we expect to provide during the project. And looking at the timeline, so in 2025, we already consolidate our regional clusters and MATs. Now we are at the stage of selecting and prioritizing the BMPs across Europe. We aim to select 20 best management practices by end of November. So 2027, we will demonstrate these BMPs and discuss with our MAT stakeholders and see how to implement these BMPs across Europe. So yeah, and we also foresee more capacity building activities in 2028 to demonstrate our outcomes from the project. Well, I have been talking about best management practices, but what are they? So in our project, we have developed a website mascot that can answer our question. You can go to the website. So on Empower website, you can see at the right side corner, ask Boy. So Boy is our mascot. You can see here, there's an option of you can chat with Boy. What you would expect from the project. You can see the Empower solutions, and here is the question and answer to what are the best management practices. In short, these are...
Hongzhen?
Yeah.
I think when you clicked Ask Paui, maybe you open a second window because we're not seeing any. I just see that's still the initial page of the...
So you don't see the Paui?
No, the box, no.
Okay, sorry. Thank you for figure out.
Yeah, no worries. Now it's coming. Now I see it's coming.
Maybe it's because of the internet. It's a bit stuck. Yeah. Okay, no problem. I will share this link in the chat. If you are interested, of course, you are very welcome to follow our website. Yeah. Okay, this is just let you know that we have this option that you can ask our Paui to get to know our project. So I hope you can see now my slides.
Yeah.
I continue. Okay. Yeah. Okay. The best management practice, in short, is effective approach to manage nitrogen and phosphorus, reduce nitrogen and phosphorus emissions to the environment, and promote circular nitrogen phosphorus use. So we have, as I mentioned, we have 5 key emitting sectors. And for each of the emitting sector, we are going to organize a workshop with our stakeholders and discuss the BMPs. Today, our focus will be the second sector, other primary sectors, including agriculture, forest, grassland, and mining. And there we have already 4 best management practices pre-identified within our consortium. So as you can read, BMP 9, it's aquaculture effluent management. 10 is greenhouse gas mitigation in forest. 11, greenhouse gas mitigation in grassland, and 12 is mining effluent management. Well, for this BMP, all of them we have a database to collect informations about what sector it relates to, what's the objective of the BMP, what's the short description of the activities that you can do with the BMP, how is the adoption feasibility, how is aligned with the current EU policies, and if it can be scaled up, if there is previous evidence of replication. Based on this information, our project developed EU replicability score that can show how can we implement this BMP across EU. And there are also indications about potential for nitrogen phosphorus reduction or circularity, current degree of implementation, if there is potential barriers or additional comments on the use of these BMPs. Okay, come back to the focus of today, also linked to the topic of other speakers. We focus mainly on the aquaculture of the other primary sectors. That means we try to recover protein from agro waste and then make use of it in aquaculture management. So here I give some examples of our previous study that's related to aquaculture effluent management. The first one is an example from Lemna Pro, which is done by our colleague. She also works partially for nutrient budget project, and in her study, the initial effort is from the pig farm. They use pig farm effluent to cultivate duckweed, and then use the duckweed as an alternative protein source for animal feed, which close the loop, nutrient loop. Also, the effluent from aquaculture can be used as a mattress for this duckweed cultivation, and our colleague already performed cultivation experiments and to check the productivity of this system over a year. If you are interested, you can read some of the papers that she published. Another example is from Sea to Land project. That's over there. The main purpose is to make use of the fishery waste and produce bio-based fertilizer. These bio-based fertilizers are characterized as also high protein, high organic nitrogen content. When it applied to the crop, it shows no significant difference compared to the synthetic fertilizer, which can be a very good alternative fertilizer and to promote our nutrient circularity within the whole agriculture system, agro-food system. So there are different ways of recovering the fertilizers from fishery waste. I won't go into details here, but yeah, this project just recently finished last year, so there are quite some reports, publications. If you are interested, please welcome to visit their website. And the last example I want to present here is from grass to algae operational group. Over there, the researchers, Masala, she did the microalgae cultivation from grass juice. This is also a very good link to another project that they are going to present during the seminar. It's the ReJuice project. So over there, in grass to algae project, The main substrate is the farm age grass that's not so qualified for animal feed or for other use, are usually treated as waste. But if we first separate them into grass fiber and grass juice, the grass juice can be a good medium for microalgae cultivation. And then the cultivated microalgae biomass can be used as alternative protein source for animal feed. So yeah, like I mentioned from the beginning, we want to get some more ideas about best management practices in aquaculture and also in agro waste valorization. So if you have experience or you have some questions regarding these BMPs, please welcome to contact us, or welcome to answer our survey there, that you can provide your experience on BMPs, and or if you have any need to include some innovative BMPs in our database, we are very grateful for your input. I will also put the link in the chat so that you can fill in your inputs during or after the seminar. So it will be valid until end of this month. Okay, by the end, I would like to also invite you to visit our website. If you have information or knowledge about BMPs and you would like to help us to build the BMP database, please visit our website here and submit your information. And please also feel free to ask Bowie if you need any help about our project. And last but not least, welcome to follow us either through this LinkedIn or through our newsletter. We would like to have you updated about our approach. Thank you.
Thank you, Hongzhen. For the moment, I don't see questions in the chat, so I will ask a question and let's see if I get the ball rolling. But I was just wondering, for the BMPs, then you would like them to already have been tested, at least in pilot scale. So it needs to be a proven practice.
Yes, that's the best. That's like I mentioned here in this slide. Am I still sharing?
No.
No. Okay. Well, so in our database, we try to include those more implemented BMPs, so that we don't have doubts about the impact, the efficiency. But so we will have an evaluation over the previous evidence of this BMP implementation. That means now we are collecting different type of BMPs. Eventually, we will scroll down and then prioritize 20 of them. If you see a BMP that has quite some potential, but it's probably not so much demonstrated yet, if it's very outstanding in other aspects, eventually these BMPs can be also selected in our database and present to the public.
Okay, no, that's very clear. And then the project will select these most promising ones, and then they will demonstrate in different regions. That's the point, right? To see that indeed it can be as effective in several different environments.
Yeah. So when we say when we say demonstration of the BMPs, for the project's perspective, we may not be able to do experiments and confer the effects. But the main purpose of the MAT, the multi-actor transition groups, is to gather experts in this area and based on their feedback and discussion to validate these BMPs if they really have that potential. So that's the demonstration will be more at this perspective.
Okay, so it's a more theoretical exercise.
Yeah.
Yeah, then it's even more important that the BMP selected have already been demonstrated in a real scenario. Okay, no, that's very clear. I don't see any more questions in the chat, so I will thank you once more, Hongzhen. But yeah, for the participants, if something pops up in your mind for Hongzhen, just put it in the chat. She will be here until the end, so she can answer. And I will move forward with our program, and I'll invite Floris Schoeters to present his activities in the ReJuice project.
I hope you see my screen.
Yes, I do.
Okay.
But so now I do not see you.
Now you should see me. Okay.
Yes.
Okay, so I will talk about the growth of a microalgae in flower juice, more specifically chrysanthemum juice. So the main question, of course, is quite simple. Can you grow microalgae on flower juice? Well, the answer is simply yes, you can. This is a draft of how we went to it. So chrysanthemum juice is made from chrysanthemum flowers. It's not specifically for this type of flower, it's with every type of flower. Once you harvest your flowers, you have a lot of waste. You have flowers that are inferior in quality, you have the stems, you have bad leaves. Usually you trash them, you compost them, or you put them in a digester. But if you press them, you can create a juice, and you have a fiber fraction. The fiber fraction can, for example, be used to make insulation panels. But the juice is actually quite nutrient rich. It contains all the nutrients except phosphate to grow algae. So what we did was, we got the juice, we store it for a long time. I will explain why you have to store it for a long time soon. And then we did a lab scale experiment just to see, can you actually grow algae in the juice? And also to figure out what kind of algae will grow in the juice. And then we went to a pilot scale test in a flat plate panel reactor of about 350 liters. And then we also analyzed the biomass to see if it was comparable with normally grown algae in mineral medium. So in February, we got the flower juice, and it was actually quite clean. And when I say clean, I don't mean it's sterile, but if you look at the left picture, you see there was no fungal layer on top of the juice, so it was quite clean. What we noticed was that the container, when it arrived, was bulking. There was a lot of gas formation. This was because of the transport. You shake it and gas gets, it's contained in the container in the biomass. At the bottom, you still have some precipitation that contains the gas. But of course, by transporting it, you move it and the gas tries to escape. Why is this important? Well, if you ever work with this type of juices, you have to keep this in mind to make sure that your container will not explode during transport. Did it stay clean? Well, no. At some point, you start to grow a fungal layer on top. But there's one caveat here. Why was it growing on top of the container? The first container we received was actually a container that you had to open from the top to take out samples. So air gets sucked in, and instead of a fermentation, you get air in the container and you get fungal growth on top of your container. Is this bad? Oh, it's dirty, but for the algae, it didn't matter. I will explain soon why. At a certain stage, you reach a steady state status where the fungi actually disappear. They die. You will see some fungi from the wall of the vessel that will drop in the liquid. But in general, the liquid becomes quite clear. Of course, again, it's not sterile, but it's quite clean. And this is what you want. Now, it's not just about the fungi. What is actually the most important thing, and this is often not studied in most experiments. Usually what you do is you get a small sample and you put it in the freezer to keep it the way as it was when you received it. But it's actually not the way to go. So one of the most heard comments about using waste streams is, well, waste streams are not stable, they differ. To an extent, this is true. However, if you have waste streams in bulk containers or in bulk volumes, large volumes, and you store them for a long time, you will see a few things changing. Now, we looked, of course, only in nitrogen and phosphorus because of the algae. We knew that it contained all the micronutrients needed for the algae, so that was not our concern. But the main message here is the nitrate, which is quite high at the start, will go to 0. This is because of the bacteria that will use the nitrate or the fungi. Is this a problem? No. Why? Because you will start to form ammoniacal nitrogen. And this is also a source of nitrogen that algae can use, of course, depending on the species. But it works just as fine as nitrate. So nitrate goes down to 0, ammoniacal nitrogen rises. It goes up to about 1,700, and it stays quite stable after about 100 days. And this is the point that you need to reach. You need to reach a type of steady state that you have a container with a certain volume that stays at around the same range in terms of nitrogen. And this is what you reach only after about 100 days. And we have done this multiple times with multiple containers, and it always takes about 100 days. Of course, it can be once 80 days, maybe 110 days, but around the 100 days, you will reach a steady state. What you will also see is that the phosphate goes down to 0. The phosphate itself, contrary to the nitrogen, is not necessarily gone, but it's taken up by the bacteria and your fungi, but it's less available for your algae. But ultimately, it's still there. So what we did was, after 101 days, we took some of the juice and we started to grow our microalgae. So as mentioned, there is no nitrate left and the phosphate is very low. It's contained in microorganisms, but it's less available for your algae. We also measured the total scale-down nitrogen, just to give a more complete view of the nitrogen. To be 100% correct, you could also measure the nitrate, but yeah, it's not that important because ultimately you know that it's ammoniacal nitrogen that will be formed and that you will take into account when you calculate how much juice you need to grow your algae. So as I already mentioned, the micronutrients, they are present in enough volumes, concentration, so there's no issue there. The main issue is the phosphate, which is quite low. Well, if you look at the table, you have to look at what's between the brackets. So we did a calculation. How do we end up with 12 liters? That's because of the nitrogen. So we calculate the volume that we need based on the nitrogen. In this case, it's the ammoniacal nitrogen. If you look at ammoniacal nitrogen, you see it's 53.5 or 0.49. Mineral medium is 56, so that's the same. Why it's not exactly the same, just do measuring errors, but it's the same amount of nitrogen. You will see that the phosphate is only 4.5, which is a lot lower than 21. So the phosphate is actually the limiting factor in this medium. Now, when you work with flower juice, you work with a waste stream and your algae are not used to this. So the first 2 days, what you get is an adaptation phase. It's actually stress. You can see it in the tubes. If you go from left to right, you see the number or the digit is the day. So day 0, day 1, morning, day 1, afternoon. Day 2, morning, the color changed. It became, pale orange. This is the stress of the algae, but they recuperate. But it takes 2 to 3 days. This is something you don't want, but we always saw this. You have to take into account that here the algae were grown in the lab in mineral medium. What we did notice is we saw an adaptation of the algae that they recuperate faster. So at a certain point, your algae get used to the flower juice and they recuperate and they don't have this long lag phase. But what we especially noticed was that the dry weight only went to about 0.5 grams per liter. And this was because of the phosphorus. Just to give you an insight about the stress. So you see actually the color, it's really, it's an ugly color. It's not a healthy culture. So at first you fear, okay, your algae are going to crash, but they didn't. They recuperated and they grew. And ultimately you end up with a nice green type of algae. Here you see the final harvest. You see it's a nice green color for this algal species. So how did we do it? We used a flat plate panel reactor and we grew 7 days. The first 2 periods. Why 7 days? Well, at 7 days, we reached about 0.5 gram and we knew this was the max. In the third growth period, it only took 5 days to reach this, and in the fourth, it only took 4 days. So you see the algae, they are adapting. The mineral medium positive control, we also did 7 days just to check, compare. And here you actually see this very nicely. So the first period, you see the first 2 days, or maybe even 3 days, you see that there is a lag phase. It's not growing. After 4 days, it starts to grow, and it reaches a certain cell. I put cells, not dry weight, a cell number, and you see that the cell number, the height of the cell number or the top of the cell number actually corresponds with the ammoniacal nitrogen that you see in the gray bar. So at some point, we don't know why this is true, but at some point you reach about 10 milligram per liter of ammoniacal nitrogen, and then the algae actually stop growing. So this is somewhat of a threshold. They need more than this 10 milligram per liter. But of course, there is also the phosphate limitation, but it always mimicked very nicely the top of your cell count, matched the minimal value of around 10 milligram per liter of ammoniacal nitrogen. And you also see in the growth periods that the days become shorter. So the lag phase that you had in the first 2 periods, in period 3, actually, there was no more lag phase. After 1 day, immediately the algae recuperated. Period 4, it was the same, no more lag phase. And that's an important indication that your algae adapt to the medium. And you actually see this even nicer when you check the pH. So the pH pattern, you see in the first 2 days, period 1 and 2, that you have a very strange pH profile. So the algae, they are not really doing much, they are just adapting, but they are not really growing. But after a certain moment, about 3.5 days in the first period, let's say 2.5 days in the second period, but immediately day 1 in the third and the fourth period, you see that the pH rises. And then you see this nice pattern of up and down. And this is actually just the system. We injected CO2, carbon dioxide based on pH. And when you reach this up and the pH that's leveling between those 2 values, then you know the algae are growing well and very happy. And you see that this is faster in period 3 and 4. So here you see the content of the algae. It's comparable with the mineral medium. There is no real difference. There are some differences, but it's more due to measuring errors. Crude fiber was a bit higher, but in general, you reach a very nice protein content. You see that the protein content was a bit lower compared to the mineral medium, but again, this was also partly due to the phosphate stress. Can you reach higher values? Absolutely. We did a winter test. This was a very basic test. We didn't optimize anything. We reached 1.2 grams approximately. We're doing a new test now, and we are already at 2.5 grams per liter actually. So if you add phosphate, your algae are very happy, and they actually grow better than in mineral medium. This is an example. How does the culture look? It has a very healthy green color. Interestingly, you have a lot of foam. Now, the foam formation when you cultivate algae is usually a sign of stress. Here, this is not the case. Here, the foam is formed because of the chrysanthemum juice. The flower juice has some tensioactive substances and forms foam. If you look at the reactor, and maybe it's hard to see, but if you look at the tubes, you do not see any biofilm formation. This is very strange to see because normally you will always see a little bit of biofilm formation, especially after 30 days of growth. But this is not the case here. So there seems to be some kind of tensioactive substance in the flower juice that prevents biofilm formation, which is quite good. But at the same time, you also have the foam. Now, the foam as such is not an issue because it's not a sign of stress, but this is something to take into account. We already are harvesting about 1/3 of the culture every week, which is quite a lot. So the growth is very, very nice. I already mentioned this. The first barrel we got with the juice, we had to open it. Air will be sucked in, which is not so good for fungal growth. So what you do is you use an airlock. It's better to keep your sample clean and you don't have the fungal growth and your juice stays relatively cleaner. But it's not necessary, but it can help. If you close a barrel, of course, you also risk contamination. But for the algae, it doesn't seem to have an effect. But the very strange thing that we noticed is when you have a barrel with an airlock, you have a separation of liquid. I have no clue what it is or why it is. It's a bit of the bottom part that you see on the left tube. It's not precipitation. The precipitation is really completely at the bottom. There is no real precipitation. It's more differentiation in the phases of the liquid. We have no clue why, but it is something we noticed. Does it make a difference in terms of growth for the algae? No. The only thing you have to do is you have to store the juice long enough to stabilize it. If you want to know more, we published the paper together with the consortium. This was the small-scale experiment and the pilot-scale experiment without phosphorus addition. If you do add phosphorus, your algae will grow in the chrysanthemum juice the same way as in the mineral medium, and actually even better. So in theory, you could easily use chrysanthemum juice or flower juice as a cultivation medium for algae, of course, depending on the species, but it works. From a legal viewpoint, of course, yeah, there are some questions to be asked, but it works. So that was what I wanted to show you. And then thank you for your attention.
Yeah, thanks, Floris. I see there are already questions in the chat, so let's go for these. Liliana is asking, if that microalgae production facility is in Belgium, is it possible to visit it? Well, I know the answer, but you can answer. But also, what method did you use to harvest the algae?
Yeah, you can visit it. If you send me an email, we can set something up. How did we harvest it? Well, it depends. If we do let's say a small scale harvest, and by small scale I mean 100 liter, we use a centrifuge. If we go higher, then we use a combination of a centrifuge and a filtration system. Then we do a first filtration system. It's called a Licoflux, where you will have a dead volume or an end volume of about 40 liters. So the more you put in the system, the more it will up concentrate. So if you go from 400 liters to 40 liters, it's a factor 10. If you put more in, then the factor will become bigger. Of course, there is a limitation, but it's a combination of filtration and centrifugation. And it can go quite fast, but of course you have to optimize it depending on an algal species.
Okay, and Boham is asking, regarding storage time versus concentration, the pH was continuously increasing from 5 to almost 9. Why this trend? Yeah,
so this is normal. So in the beginning, you have the spontaneous fermentation. The pH will drop. After a while, and this is why the long-term storage is really important. After a while, fermentation will also stop because there is nothing anymore to ferment. The food for the algae, for the bacteria or the fungi is used, and you will reach a type of steady state and nothing much will change. But what happens is, because of the fermentation stops, you will have an increase in the pH. Now, I forgot to mention this, but this is actually a very interesting thing. We don't know yet why this is, but in the flower juice, there is also something that's buffering the pH. So what happens is, if you start growing your algae, the pH will stay quite high, which is quite strange. But this has some influences on the pH regulation with your carbon dioxide. So you inject carbon dioxide based on the pH. Normally you set a pH of, let's say, 7, 7.5. You can't do this with the flower juice. You have to use a higher pH value, because otherwise you will keep injecting carbon dioxide non-stop. There is something buffering in the juice. But this is an advantage, because ultimately you need less carbon dioxide, which makes it cheaper. And the second thing, and this is really important, you are actually growing your algae somewhat mixotrophic. And the funny thing is, because the juice has a high pH, and depends on the algal species, but this algal species has a tolerance for a high pH, you can stop giving them carbon dioxide 1 or 2 days before harvesting, and you will see that your culture reaches a pH of 11, even higher. And why is this interesting? Because you will kill your bacteria. This is also important because we do not sterilize the juice. In the next test we could do this, but we tried to minimally, to not do anything, just to see what the quality would be. And it actually works quite well. Is it sterile? Of course not, but it's a quite clean culture. If you are going for food applications, then maybe you have to sterilize it or filter it, but we are not yet there. We wanted to test the most basic situation. Of course, keep in mind, it depends on the algal species. And maybe this is important. It will not work for every algal species.
Yeah, that's clear. I'll wait to see if more questions pop. I have a couple of questions as well. Well, you said, you don't filter, but the bacteria load is quite low. But did you measure the microbial contaminants in the produced biomass or not yet?
We are doing this, but in the paper you already find some answers. So ironically, and this is something that's often underestimated, It's often stated that algae cultivation, that they do it accenically. It's not really true.
Yeah.
The irony is, once you harvest your algae, you will also up-concentrate your bacteria, because you also centrifuge your bacteria, so it will rise. But of course, when you heat treat your algae, there should not be a problem, as long as you do not have pathogenic bacteria. And of course, if you do filter your juice prior to using it, you can also lower the contamination. So technically, there is no issue to do this. You could perfectly, like certain companies do, they filter it, and some even pasteurize the medium before using it. So technically, there is no issue not to do it. But we wanted to use the most cheapest test.
No, that's clear. And maybe a question then to link with the NPower project, which is the reason why we're all here today. Did you already think how this technology would be transferable? So, okay, we're doing it in Belgium. It's quite a mild country in terms of temperature, but you did the storage. I think it was outdoors. How would this transfer to a different country? Would they have to do it differently? Yeah, how do you see it?
No. So, of course, depending on the temperature, there will be differences. But the irony is, if you are in a warmer climate, you will have the fermentation a lot faster. There will be some differences, but after a while, you will reach the steady state no matter what. What you do have, if you are in a warmer climate, you will have a faster fermentation at the start, and you will have more carbon dioxide formed or gases formed, so you have to make sure that they can escape. And also, we stored the containers outside for over a year now, and we measured the temperature in the containers, and it went from, well, not freezing, but let's say 5 degrees up to 50 degrees. So the range that we reached was quite high. Of course, if you are in a country where it's outside 40 or maybe 50 degrees, then the container can go up to 70, 60 degrees. I would not say I know the answer there, but I don't expect any issues, to be honest.
Okay, so just the time frame will be different, but you would expect it to also reach a steady state.
The main messages, or the main take-home message is, it's really necessary to store it for a long time in a bulk volume, large volume, and then you will have at some point a very clean, nice, stable juice that you can use. And this is the same, just coming back to the other project, grass to algae, is the same with the grass juice. Also there, you need this type of storage to stabilize it. Of course, maybe one downside is some of these juices, they can smell very bad.
Yeah, then you choose the one that is more appealing. But okay, no, that's very clear. I don't see any more questions in the chat. And we're perfectly on time, so now I will call a little break. We will be back at 3 with the other 2 project presentations. So thank you very much for the speakers so far and the participants, and see you in 10 minutes.
See you.
Hello, everyone, and welcome back. We will continue our program with James Gaffey presenting the RuralBioReFarmeries project.
Thanks, Marcella. Can you see this?
Yes.
Perfect. Thanks for the invitation. Good afternoon, everyone. So I'm going to give a case study of the RuralBioReFarmeries project. My name is James Gaffey. I'm based at Munster Technological University in Ireland, where I co-lead the Struk.bio research group, and I'm coordinator of this project called RuralBioReFarmeries, which is funded by the Circular Bio-based Europe joint undertaking. So one of our focus areas of the project is on grasslands in RuralBioReFarmeries, and grassland is a very abundant feedstock from an Irish perspective. I've highlighted we have over 50%, almost 60% of our total land area, which is grass. So it's very predominant terrestrial land use in Ireland, but right across Europe, there's a lot of grass opportunities in terms of bioeconomy opportunities. Obviously, we feed a lot of grass to cattle, but it could be a key resource for doing other things with it in addition to feeding cows, and this has been talked about already in the webinar. So currently, it's mainly being used when it's being used as a ruminant feed, but there are lots of challenges for context that the agriculture sector faces. There are a lot of emissions associated with agriculture, and particularly livestock production, which is a big challenge for farmers. In my country, in Ireland, it makes up over a third of national greenhouse gas emissions. So our farmers are under big emission reduction targets, 25% by 2030. It's higher in some other countries. Also, one kind of driver from a farming sector, and also for the potential development of grass-based bioeconomy models, we import a huge amount of protein into Europe on an annual basis. And in Ireland, we have a particular challenge. We are very protein dependent on imports. So we import about 3 million tonnes annually into Ireland. And there's currently discussions around developing an EU protein strategy to become more resilient around the area of proteins, which is another opportunity for grass bioeconomy development. And then another area which I think we're hoping to try to help with as well. We've seen over the last number of years the cost of agricultural inputs, things like fertilizer, feed, energy, increase substantially for the primary sectors, and a lot of this is dependent on the fact that we're so dependent on imports as well, and we see this again over the last number of weeks with the Iranian crisis. So being able to become more resilient with resources to create our own energy, fertilizer, feeds would be a big opportunity, and also it's an opportunity to create new product revenue streams for farmers also. So I've titled this grass, could this be Europe's untapped protein opportunity? Because within the strategy discussions around protein strategy, there's a lot of talk about lupins and beans, peas, all kinds of different alternative protein sources, but we do have a lot of grass, and that's another key opportunity. If you look at the on the top right, you'll see we have different types of feedstuffs that are going into be used as concentrates in our feed sector. And for a lot of these, they're in light gray, you have quite a lot of resilience, and we're quite self-sufficient. But in the case of soybean in particular, we're only about 3% self-sufficient in Europe. And it's a key ingredient which farmers like. It's high protein and is integrated as a feed in a lot of diets. If you look at the table below, you can see that soy is 35% in terms of protein on a dry matter basis. So it's a very attractive crop, but on a land basis, it's less interesting or less productive. It's only about 2% dry matter per hectare. So the proteins in kilograms per hectare is relatively low if we compare it to the potential of grass and clover, which is coming in substantially higher. But the challenge with the clover and grass is being able to extract the protein and make protein more available as an alternative to feeding it only to cows. And so one of the approaches that has been developed is this area of green biorefining, where you essentially press some of the protein out of the grass. Some of the protein remains in a fiber fraction, which can be fed back to cows. And then within the juice that's extracted, we call it a green juice, the remaining protein can be precipitated out and can be made available to monogastric animals who typically can't digest grass fibers. And so this becomes an available alternative feed for animals like pigs and chickens who wouldn't be able to feed on the grass. After taking the press cake and the protein concentrate out, we're left with a juice stream, which contains carbohydrates, nutrients, some of the remaining proteins, and this can be used as a feed for anaerobic digestion to produce biogas. So this is an interesting model that's being looked at in a number of countries. In Ireland, we had a project a number of years ago called Biorefine Cluster, which was starting to look at this in collaboration with some of the farmers, some of the dairy farmers in Ireland and pig farmers, to look at the potential of this, and found some really interesting results using this mobile biorefinery system. So some of the findings we've had to date from this model that I presented in the previous slide has been that when we took our press cake fiber and we did two cattle feed trials using the fiber as an alternative to silage, we found that we had similar or even higher milk yields despite reduced protein from the cows who were on the press cake diet compared with silage. And also we had reduced nitrogen and phosphorus losses in the excrement, and we've printed a couple of papers on this area. We also found that the protein that we extracted, the grass protein, we call it a leaf protein concentrate or an LPC, can replace the soybean meal in pigs. And we've looked at this in two trials in Ireland, one at a 30% replacement rate and a second trial at a 50% replacement rate, and found very good results in terms of weight gain in the case of the pigs. And we've done some life cycle assessment work comparing this to soybean meal, where we found that we had a 5 times lower carbon footprint compared with imported soy. So there's environmental benefits coming from this as well. We also did some analysis and found that the brown juice, the residual stream for the process, is a suitable feedstock for biogas to produce process or surplus energy. So maybe combining with combined heat and power and using that back within the process to reduce the energy inputs. And then importantly, we found as well that there's a number of remaining currently untapped high value opportunities to develop some more products of higher value for human nutritional applications, for example, or for biomaterial applications as well. And then importantly, I suppose the work that we've been doing in Ireland is also happening alongside lots of international research in this area, a lot of which is finding some similar findings. And I'm just highlighting some of the countries: Denmark, Netherlands, Finland, Sweden, Norway, Denmark, and Belgium. So there's a lot of countries that are starting to look at this opportunity. So I think Marcella asked to talk about some of the challenges and bottlenecks. So there's a lot of potential, and it's a really nice model, the one that we've looked at so far in terms of the 3 products coming out of it. But there's still some commercial challenges, and that's why we developed this project, RuralBioReFarmeries, is to try and improve the model that's already there. So some of the challenges include challenges around feedstock supply. Currently, in the current model, we would focus predominantly on fresh grass or grass clover, for example. But that's a feedstock that's only available during grass growing months, which can vary between about 6, 7, maybe 8 months a year at the maximum. But it leaves a lot of months where you can't process in your biorefinery if you're only dependent on that feedstock input, which presents a challenge for the viability of the green biorefinery. Also, logistically, a lot of the operating costs for the green biorefinery are related to harvesting and logistics. So you have a high moisture, low density feedstock, which is going in. So that's difficult from a transportation point of view. And also, when we're looking at the extraction of proteins, we typically are looking for fast processing in the feedstock, and also reducing the biomass variability would be important as well. And then thirdly, the economic challenge, which is probably one of the biggest. You have, very important products which are coming out, like your protein, forage, and energy sources, but you have relatively low value associated with these products. So it's currently not the most attractive business case, even if from a resource efficiency and resilience perspective, there's a lot of benefits to it. So our project, RuralBioReFarmeries, is a 4-year demonstration project funded by the Circular Bio-based Europe joint undertaking. It's a 8.7 million total budget, and we have 19 multidisciplinary partners across 3 main demonstration sites in Ireland, Denmark, and the Netherlands. But then we have a lot of other countries that are participating and bringing their expertise to the project as well. So the aim of the project is to demonstrate a first small-scale decentralized green biorefinery model, and to support its wider replication to increase local resilience, sustainability, and economic opportunities around Europe's rural grassland areas. So in terms of how we're trying to help in resolving some of the bottlenecks that exist at the moment, so one area is on the product side. So we're trying to develop a cascading biorefinery value chain, which balances between meeting farm inputs and developing additional high value materials. So we're trying to keep the good parts that we had in the original model in terms of protein feed, press cake being available for cows, production of some energy via anaerobic digestion. But we're also exploring some of these new product opportunity areas. So some of this would include this separation or fractionation of white protein or rubisco from your protein that's extracted in the juice, so that you can create a separate protein which would go into, for example, the monogastric feed market, and then a higher value protein which could potentially go as a functional protein into human nutrition. We're also looking at things like the better development of our AD process, to not just produce biogas, but also to target high value chemicals such as volatile fatty acids, so materials like butyric and caproic acid as potential ingredients in applications like flavors, animal nutritional additives, antimicrobial packaging additives, various different applications that can bring the sustainability or health benefits, but also displace potentially fossil-based materials as well. On the fiber side, we're looking at animal feed, but we're also looking at converting some of our press cake into bio-based packaging for food packaging materials. So integration of grass-based fibers in packaging applications. And then from some of the process outputs, again, you're looking at on one hand, looking at energy and nutrients, but also some of this, we're looking at the cultivation of microalgae as well as another application from this. So it's a very, very long, I suppose, and cascading value chain, which we're developing through the project. And we have some different demonstration sites, which is taking some of these and developing them further to see what are the best avenues for improving the value chain. We're also looking at trying to build a continuous annual supply chain, and trying to understand what that would look like to try and address this 6 to 8 month window for feedstock availability. So part of this is around looking at the existing feedstocks we're using, like perennial ryegrass in Ireland, and understanding how we can make that part more sustainable, for example, through the inclusion of clover or multi-species swards to bring some environmental benefits and reduce nitrogen application. But also we're looking at potential crops that we can use outside of the typical grass growing months. So here we're looking at things like beet leaves or other plant leaves. We're looking at cover crops, for example, that will be available later in the year. We're looking at silage as an alternative feed during the winter as well. So these are a number of different feedstocks that we're looking at to try and enhance and increase the possibility to process for a longer season. And finally, one of the tools that we're trying to develop is to improve the logistics in the biomass supply chain and develop a tool which helps to improve decision making, firstly on the harvesting side of things in terms of when we should be harvesting based on yields, based on protein concentration, predicting scheduling of biomass, and also optimizing the logistical routes to the facility as well. So it's a kind of data-driven tool that we're developing with smart technologies to try and improve the overall supply chain. But we're also trying to test, in addition to this, a decentralized biorefinery concept where we look at the developments of, for example, local processing of biomass in the farm, where pressed cake could be kept on the farm and juice could be transported to the centralized site, or the development of regional hubs, which would do the processing at the more regional level with the juice transported from these regional hubs. So these are the regional demonstrators within our project. We have Aarhus University in Denmark, we have Farm0C in Ireland, and we have Akeris at Wageningen University in the Netherlands. So the two sites in Denmark and the Netherlands have already been operational over a number of years. In Ireland, we're building a greenfield site, our first demonstration scale green biorefinery, which is being developed there to try and scale up our activities. I'm going to focus more on just how we're progressing on our Irish pilot and demo side of things. So we have been working and leveraging kind of national funding for the development of our infrastructure, which comes into the RuralBioReFarmeries project. So we just launched our new pilot plant at MTU, which is doing the kind of early stage medium TRL work to try and show how we can extract out our protein and produce our pressed cake and brown juice. And during the summer, we have our downstream process, which is going to be coming in, which is going to be focusing more on the extraction of the higher value products. So that's something that's coming during the summer. These are just some of the samples that we can produce. So you've got your feedstock, your pressed cake, your green juice, and then you're extracting out your green protein, your white protein, and your brown juice, which would be the substrate that would potentially go into this AD process, or in some cases a two-stage AD process, looking at the VFA production. We're also working on the development of a demonstration site currently, which is co-led by Munster Technological University with University College Dublin and the Carberry Group, who are a dairy co-op here in Ireland. So we're building this new demonstration facility on a commercial farm called Farm0C, which is a kind of flagship farm in Ireland aimed at the integration of all kinds of climate mitigation technologies. So that covers everything from grassland management to renewable energy to animal breeding and animal nutrition, and all kinds of diverse strategies. And every year the farm measures its carbon footprint and understands the impacts that these strategies are having on the baseline emissions of the farm. And so the next big development of the farm will be the launching of this green biorefinery. So this is the plant that's under construction at the moment, and we're aiming to have this in operation during the summer. So we've been steadily developing this, and this is going to be implemented, and it's where we're going to be scaling up our research that's happening at Munster Technological University and with our partners on the RuralBioReFarmeries project. So just a kind of final slide in terms of how the RuralBioReFarmeries project is helping green biorefinery development in general. So I think it's trying to address some of the key bottlenecks that exist in the commercial value chain. And I suppose we bring in particularly experiences on the commercial side from Denmark, where there's been a number of commercial plants that have experienced some of the challenges that we're trying to address in the project. It's also bringing innovations and expertise together from different European countries. I mentioned before that there's a number of EU countries that are very active in this space in developing this area around grass. So it's bringing some of these stakeholders together. So there's a lot of shared learning between the partners. It's also developing partnerships with industry partners and primary producers. So we have both within our consortium. We have farmers on the dairy farm side and the pig farm side. We have partners who are on the agri-food ingredients industry. We have partners who are from the packaging industry. So it's bringing all of these commercial players together in a new sort of value chain approach at the EU level. We're trying to address commercial challenges, and this project is kind of commercially focused. So we're starting to think about things like regulatory aspects, consumer acceptance, aside from the actual scaling of the technology and validation of products. There's that side as well. It's improving knowledge networks and supporting replication potential. So we're going to be working to replicate this model to other countries, and also we're going to be developing a training network aimed at particularly primary producers and advisors to improve knowledge around green biorefinery development. And also, I think an aim is funding innovative research that can future-proof rural communities. So trying to develop models that could potentially be taken over by the farmers in that maybe decentralized model that I talked about, or at a cooperative level, something that could be embedded within farming communities to help them to improve sustainability, but also become more competitive and take advantage of the opportunities that are there in the bioeconomy to develop new products and new business opportunities. So yeah, these are some references. So thanks for your time, and I can take questions if there's questions.
Yeah, thanks, James. There are 2 questions in the chat for now. So Thibault is asking about the feedstock supply. Is it possible to store what you're harvesting for multiple months, or does this also have problems?
So yeah, if you're working off fresh feedstock, it's not possible. So we've incorporated silage as one alternative for some of the winter production, but with silage you would be less focused on protein as an output from in terms of a product. But silage could fit in quite well in terms of the juice extraction from the silage. Still fits in very well in terms of the AD process, the novel AD processes that we're testing. So we still can add value if silage is included as a winter feedstock. On the fresh side, you're kind of relying on, so you're typically trying to process within the day. So if you're harvesting grass, you're trying to process that grass during the course of the day to extract your protein. And what we're trying to do then in that case is to try and sequence feedstocks in such a way that we can, when the grass season is coming to an end, we have other feedstocks like cover crops that are starting to come into play to allow us to still extract protein during certain parts of the year. And then when that's not possible, then shifting to silage.
Yeah, no, that's clear. We have a second question from Hongzhen. When talking about extracting protein for human consumption, do you also need to control the plant variation growing together with the clover to avoid contamination of any dangerous plants?
Yeah, you would. I mean, so at the moment there are a limited number of grass feedstocks that have managed to get through in terms of products for the going into the food market. So it's still a bit of a challenge. So you know, and not I suppose one of the things that we're kind of looking at as a project. So there's a number of crops that we're looking at by themselves in terms of potential use as crops for producing what we call high proteins for human nutrition. But what is also going to be important is kind of doing the screening on any kinds of anti-nutritional factors that are going to be associated with these proteins. And that's something that's a big challenge at the moment. And we've been talking about within our own project and with other projects around trying to collaborate with stakeholders to develop a kind of body of evidence across different grass feedstocks and what the potential is. So mapping out what are the anti-nutritional factors that could be an issue for these types of proteins coming from these types of crops, and then trying to do some screening work looking at whether these are an issue for the crop. So it's definitely something that we have to be careful about.
Okay, no, that's very interesting. I'll move along the program, so thank you very much, James. But there is one more small question in the chat. If you can answer there, that would be great. But sure. Thank you so much. I'll invite now the Agriloop project. It's a dual presentation, so we will start with Martin, and then we will have Wei Gao with a second presentation from the same project. So Martin Struk, I give you the word.
Yes, thank you. Give me a second to load the presentation.
Yeah, that's perfect.
Okay, so good afternoon, everybody. My name is Martin Struk, and I'm a postdoctoral researcher at Ghent University. Today, I would like to introduce you to the potential of grey starch waste stream for microbial protein production, which is part of the Agriloop project, which I will also talk about. I believe I don't have to introduce the potatoes to you. Fries, chips, boiled or mashed, there are many forms of potato. In Europe, around 48 million tons of potatoes are processed every year. It is too easy to imagine that amount of waste that's generated is quite huge. One of the many waste streams obtained is the grey starch, which with estimated production of around 2 million tons across Europe. What exactly is a grey starch? It's a residual starch-rich wastewater recovered after washing of peeled potatoes. It is subjected to heating above 40 degrees, hence the grey color and the name. Currently, grey starch is mostly used as a fertilizer, low-grade feed, or in a production of biogas. However, there are incentives to find new ways of producing food or feed-grade microbial protein, and the grey starch may play a role in that. And also, this is where the Agriloop project comes in, bringing together multiple universities and companies from Europe and China. The 35 partners, half academic and half private, of the International Agriloop Consortium aim to extend the agricultural production value of two major players of the global bioeconomy, Europe and China, and by upgrading underexploited residues into the high added value bioproducts. The goal of Agriloop project is to develop safe and sustainable by design bioconversion processes integrated in a cascading biorefinery approach. Wide range of agri-residues such as tomato, soy, potato, brewery waste, and many others is converted into plant and microbial protein, polyesters, and other bio-based chemicals. Resulting products can be used for food, feed, health, and material applications, especially by the farming sector. Grey starch is one such waste stream, and it is my task within the project to address its potential utilization. You might ask, why should we dedicate energy and resource to producing microbial protein? Well, it's because it can offer various benefits. For example, it can be used as a substitute in animal feed, and its production requires minimum land, around 83% less than plant and livestock farming. It has also lower water consumption. Grey starch can be combined with other substrates and used to produce microbial protein, which improves the overall versatility of the process. Another benefit is that compared to animal or plant protein, the efficiency of nitrogen use in its production is much higher, up to 43% when used as a food alternative. Lastly, greenhouse gas emissions are lower compared to traditional protein source. However, some aspects of microbial protein need to be kept in check. It is important to avoid the production of toxins and excessive amounts of RNA, which may cause harm. The nutritional profile is equally important. The amino acid composition must align with the requirements for food or feed application. To be competitive in the market, microbial protein must provide comparable or better value for money to existing products. Lastly, the protein content of the biomass must be sufficiently high to justify production, at least 30%. This might seem like a tall order. Fortunately, Cupriavidus necator can fulfill all these conditions, which is why we selected it as the best microorganism for microbial protein production. It is a facultative hemolithotrophic bacteria capable of both heterotrophic and autotrophic growth. Its amino acid composition and content are favorable and in line with the minimum requirements for food or feed. It can consume various organic acids and C1 gases, and some strains can also consume glucose. Importantly, it is generally recognized as safe for use. So, how do we convert the starch into microbial protein? The process involves several steps. Firstly, pretreatment is required. Since the gray starch delivered from the factory is very dense, it needs to be diluted. After that, the gray starch is subjected to hydrolysis and fermentation to produce organic acids. Alternative route involves using a commercial enzymatic mix to hydrolyze the gray starch into a glucose-rich syrup. In both cases, the residual solids must be separated, followed by sterilization if possible. The second step is the actual production of the gray starch. The fermented gray starch is added to the reactor together with any necessary additives such as vitamins or nitrogen source. The reactor is then inoculated with a pure culture of Cupriavidus necator. It is important to maintain the optimal conditions. Stress due to the suboptimal pH levels or starvation can greatly influence the production of microbial protein and shift the metabolism of bacteria towards producing PHA instead of the protein. Once the protein-rich biomass has been obtained, it is necessary to dewater it to recover the targeted product. This process produces fine protein-rich powder. Now the microbial protein may be used for further tests, mainly for the aquaculture feed trials, assessing its influence on growth and any other positive or negative aspects. We have covered some theory and now it's time to look at the practice. I'll show you a brief overview of the results. Before using fermented gray starch, we tested the performance of Cupriavidus necator on various individual organic acids using rapid well plate tests. The results were very encouraging, showing growth on all organic acid tested, with the best results seen with lactate, butyrate, and acetate. The protein content measured in these tests reached up to 70%, which was very promising. Based on these results, we asked our partners responsible for gray starch fermentation to adjust the process to prioritize lactic acid production. The product we got was dominated with lactic acid, followed by butyrate and acetate, having about 10 grams of COD per liter of all acids combined. When looking at the results of well plate test, we noticed that the higher concentrations of fermentate led to reduced growth rate, which was consistent with the inhibitory effects observed for the individual acids. In case of cell dry weight, the highest values were reached at 5 grams of COD per liter, while the highest protein content among all concentrations was at the undiluted sample, reaching about 70%. To get closer to our goal of large-scale protein production, next set of experiments was done in reactors. Fermented gray starch was available in limited amount at that time, and therefore we also used synthetic mixture of organic acids mimicking its composition. The initial results of batch experiments on fermented gray starch showed that it supported the growth reaching about 37% of protein content and 30% in tests with synthetic medium. We then considered several process parameters in following optimization: optimal pH, more favorable carbon to nitrogen ratio, and better aeration. However, even under optimized conditions, protein accounted for only 50% of the total biomass in best cases on the synthetic mixture. Therefore, we went for the other approach based on using hydrolyzed gray starch and the mutant strain of Cupriavidus necator capable of utilizing glucose. Our partners at Avecom were able to successfully hydrolyze gray starch, and they reached around 120 grams of COD per liter on average, with the glucose content about 80% in the mixture. With the addition of nitrogen and phosphorus source to the feed, we were able to successfully grow a new strain of Cupriavidus. In following runs, reactors were operating in fed batch mode for about 3 days with the pH kept at neutral levels and carbon to nitrogen ratio at about 5. Feeding was done in pulses for every 12 hours. At the end, we were able to reach from 9 to 12 grams per liter of biomass with improved protein content up to 73% and yields up to 0.30 grams of cell-dry weight per COD consumed. In further experiments, we would like to focus on improving those yields and also the protein content. Based on these lab-scale experiments, we prepare guidelines for scale-up, which were passed to our partners at AVECOM for pilot microbial protein production. Currently, about 20 kilograms of dried biomass with 60% protein content was produced and is being prepared for feeding trials on shrimps, which are going to start very soon. To conclude this presentation, I'd like to summarize the key points. Gray starch was found to be a viable feedstock for microbial protein production using Cupriavidus necator. 2 treatments of gray starch were tested: fermentation to organic acids and hydrolysis to sugars. In laboratory scale reactors, fermented gray starch supported up to 37% of protein content, while hydrolyzed gray starch allowed reaching up to 70% and was therefore scaled up further. Scale up to pilot level was successful, producing 20 kilograms of dried biomass with about 60% protein content, and feed trials are to be started soon, which will complete the full picture of utilization of gray starch-based stream. Lastly, I would like to thank my colleagues from Ghent University and partners from AVECOM for their help, and also I would like to thank you for listening to my talk. Also, at the end, I would like to invite you all to the conference in Barcelona, where the final results of the whole Agriloop project will be presented. And now, if you have any questions, I would be glad to answer them. Thank you again.
Yeah, thanks, Martin, so much. That was super interesting. Actually, I would like to ask Wei Gao to already present, and then I'm going to take the questions for both of you together because it's the same project, if that is fine. So, Wei, if you can share your screen and give your presentation. Yes, I see you now. Yeah, we see your presentation. You may start. I don't hear you. Yeah, no, I know that you're not muted, but for some reason the sound is not coming through.
Hello.
Yes. Hello.
Yeah, I'm here.
Perfect.
I'm Wei Gao from the Institute of Agricultural Resources and Regional Planning, Chinese Academy of Agricultural Sciences. And today I will give you a talk about mushroom protein and some of its application and some of our research progresses. I'm also working in this Agriloop project. And this is the overview. As Martin mentioned just now, and there are quite a few challenges come up with the traditional proteins, animal and plant proteins. So that's why we need to find some sustainable new protein sources. For example, microbial protein or bio-based proteins, which can be a promising and sustainable alternative. So that's why we mentioned about this mushroom protein. And so why we do mushroom protein in China. China is the largest producer and consumer of edible mushrooms. we have nearly 1,000 species of edible mushrooms, and among which 80 species have been cultivated, and more than 30 species have large-scale commercial production. In 2023, we have 43.3 million tons of the total production. So in this table, I listed the production and compared to the other main producers over the world. You can see the production here, and this pie chart shows the production in EU countries. You can see the largest producer in Poland is in Poland, and the second one is the Netherlands, but the total production is only 1.1 million tons in Europe. So China is really the largest mushroom producer and consumer over the world. So I listed about 30 species, mushroom species we produced for the, for example, the top 30 species here. I hope you know some of them. And what is the nutritional value of edible mushroom proteins? We have generally like 7% to 35% of the protein content. So compared to plant protein, we have mushroom protein. There is complete amino acid profile. For example, for some species, they are rich in lysine and sulfur-containing amino acids, and it has a high digestibility, like 70 to 80%, comparable to soil and rice. It has also contains some bioactive protein, like lectins and lacase with health benefits. And there are also some essential amino acids, which is varies by species. For example, there is high lysine content in this residual mushroom and agaricus bispers. That's the white button mushroom. So for a lot of species, there are a large amount of antioxidants in the fruiting bodies, and they are used a lot in Chinese medicine and for the health supplements. So the protein in mushrooms can be extracted with a list of methods. The most popular one is the alkaline extraction and acid precipitation method, but the extraction rate varies from 1% to 70% depending on species and method. So there are a lot of challenges for the protein extraction for mushrooms. For example, low yield and protein denaturation and co-extraction of impurities. So actually, we don't extract a lot for pure protein from mushrooms. We usually use mushrooms as a whole for food and for some food alternatives. Another problem for mushroom protein is the protein determination method. So the most commonly used one is Ghent method, but it generally overestimates the protein content due to non-protein nitrogen. And a second one method is the spectral photometric method, which is fast and low cost, but varying accuracy. And the last one is amino acid analysis, which is mostly accurate and which reflects the true protein content, but it has low efficiency and high cost. Which is needed for standardized protein determination method across laboratories. So this diagram shows the protein isolation and extraction process from edible mushrooms. But as I mentioned previously, we don't extract the protein a lot. We usually use the mushrooms as a whole. Because apart from protein, there are some other useful parts from the mushrooms. And when we talk about mushroom protein, and scientists compare the functional properties compared with plant protein. For example, it has better, higher oil holding capacity and better foaming capacity, and which can be used as meat substitutes, baked goods, and flavorings, for example. So it can be used in food industry, for example, as mushroom flavored vegetarian steaks, as tofu and in bread. It has better hardness and crispness in meat alternatives, and it can be used in food supplements to increase protein content and mineral content. It can be also used in beverage and flavor materials. So, for example, for meat, when we use as meat alternatives, it has the meat like hardness, chewiness, and it can substitute the meat without changing the meat elasticity. But when we compare to soy protein has a strong bean-like flavor, while mushroom protein has a mild taste, making it suitable for producing animal protein substitutes. And one more thing is mushroom protein is free from allergens, and allergic reactions caused by mushrooms are extremely rare in the population, and so it's a safe protein source for consumers. And apart from food alternatives, meat alternatives, mushroom protein can also used as the fungal leather alternative. You can see here shows you some of the products. So it needs to produce this kind of thick layers of mycelium and to do the processing. For example, when we cultivate the mycelium, we should maintain the carbon dioxide concentration and the relative humidity and oxygen concentration in the growing room, and to produce leather-like mycelium without fruiting bodies. So far, for mushroom protein production also has a lot of challenges. For example, the nutritional evaluation, which is lack of standardized method for digestibility and the bioavailability level, and the low extraction efficiency, and the consumer of most of is the consumer awareness, which is low compared to animal and plant protein, which has low acceptance and understanding, and it has market competition. So there are some future prospects. We can use different species to improve the protein content, or even use biotechnological approaches to enhance the protein yield, or to develop more kinds of products, and to standardize the quality control and testing standards, to educate the market. So for the next slides, I will mention some of the progresses of our project. So for our project is mainly working on the utilization of agricultural residues to high efficiently produce mushroom proteins. So for mushroom production, we can use almost all kinds of agricultural residues. So we mostly working on some of the aspects, for example, strain selection and substrate formula optimization, and enhancing the protein content by genetic engineering. So we tested I think about 27 mushroom species, and then we selected the higher mushroom producing species. Finally, we found that Biorefine like straw mushroom, and Pleurotus citronopulatus, these two species has generally higher protein content. So we finally selected this Pleurotus species as the materials for the subsequent experiment. Maybe you don't know this mushroom because this mushroom is usually produced in greenhouses in China, so which doesn't have a generally climate controlled growing environment. So that's why we selected this the second one, Pleurotus, this species, as the materials. And we use both GEDAL and amino acid method. We use total amino acids subtracted by free amino acid. So we can see the significant correlation between the two, between the data of the two methods. And for this species, we selected strains from this species. So about 18 strains we tested the protein content of the mycelium, and then we selected this strain as the material we are doing for the genetic engineering. And we tested the protein content and the nitrogen utilization rate, and we also see the significant correlation. According to the study in plant and other microorganisms, we selected this gene as the we think this gene is the key gene that influencing, controlling the protein production in mushrooms in this species. So this is the asparagine and synthesis genes. So we identify the genes in this in the genome, and then we analyze the gene structure, and then we tested the enzyme activity, and we see in the high protein content strain there are really has higher enzyme activity and the relative expression level of this gene. So we compare the high protein strain and the low protein strain. We can see the relative expression level is consistent with the protein content. So we construct the plasmid and for gene overexpression and RNA interference. So we got the transformants of the overexpression and RNA. So you can see here the overexpression. In the overexpression transformants, we really has the higher relative expression level for this gene, and We also tested the protein content, the soluble protein content, and the enzyme activity in the transformants in the overexpression and RNA transformants. We can see the protein content of the overexpression strain was 31% and 21% higher than that of the wild type, and the soluble protein content also higher than that of the wild type strain, and also the enzyme activity. So
Wei,
apart from
can I ask you just to go a little bit faster, because we only have 5 minutes left in the session, otherwise I cannot even take any questions.
Okay. We also tested the composition analysis of the agricultural waste, and we test the mycelium growth on different mediums in order to optimize the substrate formula. And we also used pallet scale demonstrator in companies. We tested new formula, substrate formula. We has a 3.7% of the yield increase. So thank you for.
Thank you. Good, you are almost at the end. Okay, great. Thank you so much. And sorry I had to rush you a bit. It's just that I want to have an opportunity to have you and Martin answering some questions. So I'm just checking in the chat. For now, I don't see anything, but I do have a question. Yeah, Martin, I would like you to come back.
Yes, I'm here.
Yeah, great. I'll start with you. I have a question for you on the variability of the gray starch. So did you check this in the project? Is it always the same quality, or does it change with the, I don't know if different types of potato or the moment that the potato is being processed?
Yeah, there could be some changes. We did the experiments from one batch to exactly avoid this influence. But for the future, yes, there should be some seasonal or regional variation, but we haven't measured that further. Maybe there were some internal tests at our partner Avecom, which were responsible for the hydrolyzation, but if they were, this will be published in time.
Yeah, okay, no, that's clear. And maybe just a second question from my side. Okay, yeah, potatoes here in Belgium are a huge industry, so we have this gray starch quite available. But do you know if there's any other industry that produces a similar stream, or is this really, really specific for potato processing?
This stream is specific for potatoes, as the companies. One of the problems with that the stream it could be very dense or it could be quite diluted, and for companies it's unsustainable to ship with something which is basically almost water. So many times it's just being dumped because it's economically not feasible for them. So this could introduce a solution to that, I think.
Okay, no, that's very interesting. And also with this increase in precision fermentation, it's so important to have more sustainable sugar sources. So I think the gray starch can have a very important role to play there. Okay, and maybe Wei, just one question for you, and then we can wrap up the session. From the agricultural, because I had to rush you, so maybe we can come back to the last point of your presentation, the agricultural wastes that you tested. Did you see one that was the best for mushroom production, or were they performing all very similar?
You mean?
At the end, you said that you tested some different substrates coming from agricultural wastes.
You mean this one?
I don't see. Yes, that one. Yes.
Yeah, we just test the mycelium growth rate and then combine them in substrate for mushroom growing.
Okay, and I see now from the graph that there were some that were much better than the others. So the fungi the fungi is picky. It's not any substrates that they will perform equally. Okay, no, that's very interesting. Okay, well, sorry we had to rush a little bit, but thank you everybody for being here, for the speakers, for the interesting presentations, the audience for the questions in the chat. I hope it was a good session. I learned a lot, so happy. Yeah, and I hope to see you in the next sessions. I will send the material from this session later by email, also with the information for registering for the next one. And this recording will become available in the YouTube channel for the Biorefine Cluster Europe. So thank you very much, and I hope you have a nice end of your afternoon. So bye-bye.
Thank you.